Today we will discuss about the next section, that is asexual reproduction. But before going to that asexual reproduction, we should have to learn. Last day, it's a quick recap of that last day. So last day, what happened? Last day we were talking about that reproduction. Is what? Reproduction is create a replica of the parent cell. Okay, or the same type of individual is creating same type of similar type of organism, that is called reproduction. If through the reproduction, that the species should be similar but not identical. Why not identical? Because what happened through the sexual mode of reproduction, the gene is coming from the father as well as mother. One is that gene coming from female, one is coming from male. So it's coming together and while coming to that parent, to the offspring, new other character also coming to the daughter cell. So what happened? So next the variation will come, because the incorporation of the new characters within the individual, or the new characters coming from either from father, either from mother, it's together coming, creating the variation. Variation is the accumulation of new character which can overcome, or which can able to sustain the species in the adverse or non-suitable condition or unfavorable condition, that is the variation only. Now come to the next. After that, what is the variation, and what about the character? The character is coming with the help of the DNA is going to come from the parent cell to daughter cell, right? Now, what are the different modes of reproduction? The different mode of reproduction is sexual reproduction and asexual reproduction. Asexual one is the primitive one, and sexual one is the advanced one. If you so as for that, yesterday that in the last class we also discussed about the asexual mode of reproduction and sexual mode of reproduction. What are the dissimilarities of that? The asexual mode of reproduction is needs only one parent, right? And that for sexual reproduction, two parents required. For asexual reproduction, only vegetative structure is required. So no special structure has been required for that asexual mode of reproduction, and as that no other specific structure has been required for this, that means no haploid cell is involved in that asexual mode of reproduction. Only sexual mode of reproduction, it is there the haploid number of cells. So now come to that the second phase is so what is that? Asexual mode of reproduction. Asexual mode of reproduction is what? If we going through the evolutionary mode. So in evolutionary mode, what happened? First, that single cell, unicell, unicell means single cell. Then it's going to more complex, and formation is what? That multicellular organism, right? So what is that multicellular organism? Cat, dog, human, all are comes under multicellular organism. So first we went with that unicellular organism. So assume that is amoeba. Do you ever think that how the amoeba is going to increase its number? How it is its number? Do you ever think? So you know that there is some process, right? There is some process through which amoeba also can reproduce, though it is a unicellular organism. So what happened? So first thing is coming, that is fission. Fission is what now? Then fission is what? Fission is a process through which unicellular organisms, mostly the unicellular organism, is going to divide. Okay, so suppose it's an amoeba. It's an amoeba, and then what happened? That is amoeba, one cell, cell division, is happening over here, and then come to two daughter cell, right? So what happened? Please keep it in mind. Then that what happened over here? That nucleus is going to divide in this section, right? And then cytoplasm. The cytoplasm is also going to divide with the help of a plate. So here what happened? The extension of nucleus. Nucleus is known as nucleokinesis. The extension of the so then what is that nucleokinesis? That nucleokinesis is what? That extension of nucleus and the division of the nucleus, and it's going to divide over here and contributing with the two daughter cell, that is that nucleokinesis. One thing is there along not only the nucleus, right? Here the cytoplasm is also going to divide, right? The cytoplasm first is extended. The total amount of cytoplasm is extended and then it's going to divide. So the division of cytoplasm is known as cytokinesis. Okay, please do remember what is nucleokinesis and what is cytokinesis. Nucleokinesis is what? Nucleokinesis is the division of the nucleus. Extension and division of the nucleus is nucleokinesis. And then cytoplasm, the cytoplasm is going to divide and it's become a cytokinesis. Okay, now the fission. The fission process, if you think about that fission process, is of two types, right? Fission process are of two types. How many types? One is binary fission. Binary fission, one is binary fission and one is multiple fission. Now what is binary fission? If you think that binary fission is what? We've already discussed about that amoeba, right? So what happened in case of amoeba? It is going to divide and create two daughter cells, right? So it is going to create two daughter cells. And what are the different, and that type of division is what? If you think that type of division, so that it will be the transverse one, right? That transverse means it's a horizontal way. Okay, that horizontal way, that is transverse. So that is one kind of binary fission, and it is going to create two daughter cell over here. And then another thing is there, that is binary fission, which can able to find what? Leishmania. Leishmania, or its another name is what? You know, all of you know that Kala Azar, right? So what happened over here? So in case of Leishmania, if you think, so binary fission is also of two types. Why? Because one is transverse section. That binary fission in case of amoeba, that is what? That is binary fission. And then two daughter cell, that is transverse section. Transverse section. Now if you think about the Leishmania, how it looks like? It is look like this, right? Sorry for my drawing is not very good at all. Okay, so what happened there? What happened then? In case of Leishmania, the transverse section, if you do that longitudinal section, if you put a scale like a standing line, so it's become a longitudinal section. So what happened? If you remember that structure, and then that two that's longitudinal section is there, and then two daughter cell has been formed. two daughters cells has been formed in case of Leishmania, right? So that is what transverse and that is what longitudinal. Now in case of multiple, what happened? Do you know the plasmodium? The plasmodium. The plasmodium has what? That is a cell-like structure over here, right? And thus there is no transverse division, no longitudinal division. That is randomly division, and that division is known as a multiple division. So what happened? Instead of one cell, what happened over here? Instead of one cell, so many merozoid cells has been formed over here, and after a certain time when it will get the favorable condition, all of a sudden it is going to burst. Okay, it is going to burst, and then all the merozoid cells are coming outside. Okay, all the merozoid cells are coming outside. So that is known as the multiple fission only, and the example is the plasmodium. Okay. So till now that fission is clear. So fission is binary fission, and within the binary fission, amoeba has the transverse section, and the Leishmania has a longitudinal section. On the other hand, there are multiple division, and the multiple division has been found in plasmodium, where merozoids has been found, and then lots of merozoid cells are. Lots of that sac will be burst, and that lots of merozoid is coming together. Okay, so now what is that next? Next is that fragmentation. Fragmentation is what? If you think about that unicellular unicellular, now come to that multicellular. That is what? That is nothing but a Spirogyra. How you can recognize the Spirogyra, and why that is name is Spirogyra? Spirogyra, why it is named as a Spirogyra? Because of its nucleus is coming on a band-like structure, and it's a formation of spiral. Okay, so it is known as, because of the nature of the spring-like structure, its name is Spirogyra. So what happened in case of a Spirogyra? So now Spirogyra is what? Spirogyra is an algae, and algae is used to live in what? In water, right? So what happened? That algae is living in the water, and with the flow of the water, that Spirogyra scum is there. With the flow of that water, the heavy flow, okay, on the water pressure, it is going to tear. Accidentally, it is going to tear from somewhere. So fragmentation is what? Whenever that single cell is going to, because of any accident, water pressure, it is going to break from his or detach from his parent cell. Each and every cell has the capacity, to make a new cell, right? To make a new cell or create a new individual. Okay, this thing is known as what? This thing is, it is a fragment. Fragment means small pieces. If you think that is a long chain of Spirogyra, so it is a small pieces, right? That is a small pieces of it. So small piece means that fragment is nothing but called as a small piece. So that fragmentation, the small piece of a Spirogyra is going to generate each and every cell has the capacity to generate a new individual. Okay, so this thing is known as Spirogyra, and that is the fragmentation of Spirogyra, and Spirogyra is nothing but an algae. Now come to that algae done, right? So now it's, do you ever know, okay, one more thing is that, now come to that, do you ever notice a bread piece? A piece of bread is there, right? A very small piece of bread is there, and after a certain time, you can notice that some blue-green patches are there, right? Some greenish or reddish patches are there. Okay, so what happened? What are those? Those are nothing but bread mold. What is a bread mold now? That is growing on a bread, so it is come on bread mold, right? So what is the bread? That bread molds are nothing but a fungus. That fungus is known as rhizopus. Rhizopus. Why I was telling about that that is a bread and it has different colors. Why I was telling all those things to you? Because if you notice that Rhizopus, a spore formation, that Rhizopus is a fungus and its reproductive mode is the spore formation. Right? That is a spore formation or spore forming forming organism. Spore formation or spore forming. So what happened over here? So as it is a fungus, so what happened, if you remember your lower class studies, you can know that that fungus has the capacity that it can able to take nutrition from the dead and decaying material, or that like if anything from the outside, right? You already know this. So the thing is that if you think about the Rhizopus, so that is also a fungus and it has the capacity to take nutrition from the piece of mold, piece of bread, right? So they are so tiny in structure, so we can't even see them properly. But the thing is that while changing the color, we can observe the changing in color of that mold section. So what happened? First thing, it has a hole first. It has a hyphon and it has what? That is so nothing but a sporangium. Sporangium. That is hyphae. And so what happened? And that is also known as sporangiophore because it is holding the sporangium, right? So what happened? Initially it is in different color, but whenever the spore is going to mature, so the color is also going to change, okay? And after a certain time, what happened? The lots of spore, that sporangium is a sac-like structure, a bubble-like structure, okay, that can able to hold the, or that can able to protect the spore from the outer environment, or even from outside and from the unfavorable condition also. And whenever it is going to get that suitable environment, so what happened? The sporangium is going to burst, and what happened? That rest of the spores are coming out, okay? The spores are coming, that spores, the numerous spores together is coming out, and then it is going to fall on other bread or other moisture thing, and then it is going to start somewhere, okay? With the help of this, that they can proliferate themselves in the through that asexual mode of reproduction, that is spore formation, and that is all about the structure of Rhizopus. Okay, so now we have the spore formation and it happens only in fungus, right? Now we come to that budding. Okay, we come to that next, that is budding. What is that budding now? Bud you already know, right? Anyone can tell me that what is budding? Yes, you all know, right? Bud. Bud means what? Flower bud, right? We used to tell about that flower bud. Flower bud means what? That before happening in a bud, it's a very small tiny structure and it's a rounding shape, right? So that structure which has the capacity to generate a flower. So that is the budding which can able to found. So what happened in case of, if you think about the yeast, Saccharomyces, if you think about the cell, so what they can able to do, with the help of the budding, they can proliferate themselves. Okay, so what happened if, if that is, for example, Hydra, that is yeast. So what happened? If you notice the Hydra is how Hydra is look like? Sorry, yes, my drawing is not that much good. Okay, tentacles are there. So what happened? So small flower, small bud-like projections are coming out from hydra, from the main parent body. And then what happened? Whenever the So what happened? It is going to detach from the parent cell and creating a new hydra. Okay, new individual. So what happened in case of hydra? In case of hydra, what happened in case of hydra? Their small bud-like projections is coming out from the parent body, and whenever the bud-like structure is mature enough, and it can able to do everything, every work necessary to survival shape, so it is going to detach from the parent body and acting as a new individual, that is called as budding. Not only in hydra, there is other organism also. See, if you notice that yeast, I wrote that example yeast over here. So if you think about that yeast cell, small outgrowth is there, and then that small outgrowth is going to detach from the parent cell, and it's creating two daughter cells over here. With the help of that budding, that hydra and yeast can able to proliferate, that can able to proliferate their own daughters. Okay, so through the help of the budding, they can able to reproduce there. So next day we will talk about that regeneration. Okay, next step we will talk about the regeneration and then vegetative propagation. Next step we will talk about what? Regeneration and vegetative propagation. Not only vegetative propagation, within the vegetative propagation, tissue culture. Tissue culture, we will also discuss about the tissue culture. Okay, so and after that we will discuss about the sexual mode of reproduction. So